So hopefully you can see my screen now.
Yes, we can.
Yes, but we can't see the, we see the presenter view rather than the presentation view.
Oh,
Yeah,
that's
that's
fine.
great.
Okay, good afternoon, everyone. So my name is Amr. I work for Kingston University, and today I'm going to brief you about the use of contact lenses for ocular drug delivery. So I'm going to start with a bit of a background and then have some kind of studies that we did at Kingston, and also going to look at where are we in terms of the use of contact lenses to deliver the drugs into the eyes. So contact lenses was initially used in order to do sight correction, so for myopia, hyperopia, and then patients who have stigmatism. Until the 1960s, where they discovered a monomer, which has or a hydrogel, we call it, because it has a high ability to absorb high percentage of water. And this was known as 2-hydroxyethyl methacrylate, or for short, we call it as HEMA. And this was discovered back in 1960s. And then since then, we started to see soft contact lenses or contact lenses that can be used on a daily basis. And this become more convenient to people or users because they are less rigid and then they are user-friendly compared to the hard contact lenses. The good thing about these hydrogels is they can get cross-linked while we use ethyl glycerol derivatives, for example, ethylene glycol dimethacrylate or ethylene glycol methacrylate and so on. So we can cross-link the monomer, and then you end up having the soft contact lenses that can be used. And then over the years, the more monomer has been developed. Some of them become kind of more oxygen permeable, and then we call them siloxane-based or silane-based polymers, such as TRIS or BDMS or FSA and so on. So for us as a formulation scientist, as Raj has mentioned earlier, we are interested in coming up with a delivery system that can help us deliver small or large molecules into the patients. In this case, for example, is eye. A first point that we need to think of is how we are going to build up this system. Okay, how we are going to put the drug into this system. So there are different approaches that has been investigated over the last few years. And one of them starting from a very simple approach, which is called soaking approach. So basically you use a commercial available contact lens, and then you soak them into a drug solution. So we assume that some of the drug will diffuse into the contact lens. And then you can give it to the patient, and then the patient can use it. But one of the issues of the soaking approach is there is a limitation in terms of how much of the drug you can load into the contact lens. Usually there are a small drug loading that can happen, and this will depend on the nature of the drug, if the drug is hydrophilic or the drug is hydrophobic, and again, the nature of the monomer that have been used in preparing the contact lenses. And then over the time, they came up with approaches like particle-laden contact lenses, and then molecular imprinting, and then I'm going to cover them in more details in this diagram. So I started working on contact lenses around 2014, so almost like 7 years now. And then when we started, we thought of doing a review to see what is available there and so on. So contact lenses, you can load it with the drug, as we said, through soaking, or you can put some nanoparticles on the matrix of the contact lens, and then these nanoparticles will hold the drug within their core. And as a result of this, the drug will have to diffuse through the nanoparticle that you have. And then after diffusing into the contact lens matrix, it will diffuse into the eye and then start having a therapeutic effect. Or what we can do is we can have liposomes. And then the good thing about liposomes, it can take different types of drugs. So if the drug is water soluble, you can put it inside in the core where you have your polar head of the building units, or you can, if you have your drug which is water insoluble, you can have them along the aliphatic chains that you have in your liposomes. Another approach that again was explored in order to sustain the release of the drug across the contact lenses is use of cyclodextrins. So cyclodextrins are cyclic structures, and that they are able to host our drug molecules, and they are versatile, so they can host a different range of drug molecules with different characteristics or size. And then they can improve the stability of these drug molecules, and in the same time they can elongate the drug release. So it was explored as well, and then alpha or beta or gamma cyclodextrins can be used, have a drug in there, and then incorporate your drug inside the contact lens. A fourth approach that was explored as well was the use of surfactants. And as you know, surfactant will have a hydrophilic head, and then the aliphatic chain, which is the lipophilic tail. And this surfactant, they found that when they incorporate in the contact lens, it will sustain or elongate the drug release across the lens. Before I start talking about these studies, why contact lenses will be a good or a potential drug delivery system or vehicle for ocular drugs. Basically because there are a number of issues with the current available formulations. So one of them is eye drops, for example. They are might be easy to use, but the drug available through the eye drops is very small. So we're talking about 5 to 10% only of the drug that will be able to reach the eye. And then the remaining 90% or so will be lost because of the washing out by the tear fluid, or because of lacriminal drainage, and then some of them will be swollen and so on. Also, if we think about ointment or eye cream, so again, they affect the vision of the patients, and then the adherence of this medication might not be very good. Also, I think across the previous session, we have learned about injections into the eye, and then we learned that majority of the patient wouldn't like this option because it is invasive, and then there are a number of issues, for example, contamination or detachment of the cornea and so on. There are a number of challenges. However, with contact lenses, using them that will overcome a number of these issues, and then they can be a potential solution for delivering the drugs into the eye. Also, it was quite interesting to hear that some pharmaceutical companies, for example, Novartis and Google, I think like 8 years ago, they wanted to use contact lenses to measure the amount of glucose and then use it with diabetic patient to kind of sense the amount of glucose in the blood, because they found that there is a relationship between the amount of glucose in the tear fluid and reflecting the glucose in the blood. So that was an initiative, but later on they said that is not going to work and so on. One thing, so as a formulation scientist, I'm interested in coming up with the delivery system. So I started by developing nanoparticle leading contact lenses, and we tried to use it to deliver a glucocorticoid drug, which was prednisolone. So we started this with my PhD student, Shilan Mustafa, back in 2015. And what we did is we had prednisolone, and then we started using PLGA or polylactic co-glycolic acid as a polymer. And then we, after mixing them together, we started adding them through titration into polyvinyl alcohol, which acts as a stabilizer for your nanoparticle. And then once you prepare nanoparticle, you start adding them to the matrix of your contact lens. And this will take me to the following slide, which is manufacturing of the contact lens. So basically, we use the HEMA polymer or monomer, and then together or by itself or together with some silicon-based polymer. And then we start adding some kind of cross-linker, and then photoinitiator if you are going to cure this polymer under UV light, or you can cure it with the using temperature, so you can leave it under UV for 3 hours, or for example, or you can leave it on the oven again for a few hours, and then we'll be able to get a cured contact lens. If we decided to add the different kind of sustaining release drug approaches that I discussed earlier, for example, cyclodextrins or the nanoparticles and so on, what you do is you add it at this stage when you, after you bit out your polymer together with the cross-linker and so on, you start adding your nanoparticles into the system, and then you cure it, and then you'll have your lens with the nanoparticles embedded in this lens. Okay, and then after we design the system, we need to look at this, the drug, and then characterize the nanoparticles prepared. So you look at the morphology using a scanning electron microscope. You're trying to collate some data or information about the size using the data analyzer, for example, and so on. Also, we need to learn about the state of the drug inside these nanoparticles. Is the drug still in the crystalline state or amorphous state? And we do this through using some thermal analysis technique. For example, if we look at the PRID thermograph, they're using the differential scanning calorimetry, we can see that the drug, the prednisolone, the drug has a melting point at around 250. But once we added the drug into the nanoparticle and in the contact lens matrix, we can see that in the green line there, or the green thermographs there, we wouldn't have any melting point. So this means that the drug has changed from a crystalline form into amorphous form by getting it solubilized within the nanoparticles and during the process. Okay, so because contact lenses will be applied to the eye and then be used by the patients, we need to make sure that these lenses are suitable to be used. And as a result of this, there are a number of mechanical testing and then characterization that we have to do. So one of them, for example, is hydration. So how much water will these contact lenses will be able to take once they are applied to the eye? And this is quite important because if the hydrogel or the lens doesn't take much water from the eye or didn't get wet, so this will result in the patient will feel discomfort, and then they will start feeling that there is a solid body in their eye, and then they wouldn't be able to adhere to taking it, and then it will cause issues for the patient, and they wouldn't be willing to use it in the future. So what we do is usually we work out the dry weight of the contact lens, and then we immerse it in a tear fluid solution and see how much water has been taken up by these contact lenses. In this case, for example, our control has taken around 46% of tear fluid into the lens, and then compare it to the medicated contact lens where we have our nanoparticles, one with the 0.2 gram and then another one with 0.4, with a slightly higher amount of the nanoparticles. We see that there was a slight drop in terms of the hydration percentage. Also, it's quite important to measure the transmittance of the light transmittance through these contact lenses, because of course they will put it on their eye, and then it's quite important to make sure that it is very transparent, otherwise it will affect the vision of the patient. And adding a drug or a nanoparticle there might cause an issue with that kind of light transmittance. So when we looked at the control, we had around 94% light transmittance, but this dropped by around 8% when we started adding our nanoparticle with the prednisolone. Also, as I said, the mechanical properties is quite important. For example, this will give you an idea about the handling. So when the patient takes the contact lens or the user and try to apply it to the hand, if it is very rigid, it might break. So it needs to be elastic to some extent and so on. So we used to measure Young's modulus by finding a relationship between stress and strain and so on. And then the good thing of using the nanoparticles or adding them to the contact lens, it didn't have an impact on the mechanical properties of the lens when compared to the control. So the key importance for us is to make sure that we can elongate the drug release using these contact lenses. So what we did is we had a contact lenses with some nanoparticles, and then just the nanoparticles by themselves, which contain the drug without being in the matrix of the contact lens. And as you can see, that the contact lenses manage to sustain the drug release through for longer time. So up to 24 hours, you can see that 10% of the drug has been released, and then this means that it can be used in some studies. They managed to sustain the drug release for over 2 weeks, for example, compared to the pure drug or the drug itself, or compared to the drug in a nanoparticle that can be available as well as an ocular eye drops. And this will take me to my second study. So we done and prepared some contact lenses in the lab. So now we would like to see what the patients and the healthcare professional think about using these contact lenses. Are patients willing to use it? Are healthcare professionals are willing to prescribe it or dispense it, and so on. So we did this study with a collaboration with some of our colleagues at Moorfields Hospital. And then we managed to recruit around 151 patients and then around 112 healthcare professionals. So as you can see in this table, it summarizes some of the demographics of the patients that has been recruited in the studies. And we had around 38% of the participants were males, and then 62% were females. And then more or less over 60% of the male participants were willing to use contact lenses to deliver the drugs into their eyes. And then just below 60% females were willing to use them as well. What we found interesting is the age group play important factors in terms of the acceptability of these new formulations. So, for example, younger population, 71 and 76% of younger people were willing to use it. However, when the patient become older, they are less willing to use it, and then they would like to stick to the conventional delivery systems such as eye drops and ointments. In terms of healthcare professionals, we managed to recruit some community pharmacists, hospital pharmacists, consultants, ophthalmic surgeon, and ophthalmologists as well. Okay, and then when we asked them whether they would like to prescribe or dispense the medicated contact lenses, there was kind of variation in responses. Some of them with against, and on the other hand, and some of them were willing to dispense and prescribe it. Yeah, so I think a majority of you are familiar with this table. So if you work in a hospital or a pharmacy, majority of the eye formulation or the formulation that are dispensed to the patient are eye drops. And as we can see there, 131 of the patients that were recruited have already used some kind of eye drops during their period of treatment, and this was followed by eye ointment. And when we asked them why you are willing to use contact lenses, so the patient said that simply because it is going to reduce the number of applications. So just apply the lens, and this will be once a day, rather than applying eye drops 4 or 6 times a day, which is less kind of convenient. Also, it's less time consuming, and then they felt that medication is better taken into the eye compared to the other formulations. But the people that said that were not willing to use contact lenses, they were a bit worried about damaging their eye by using these lenses. Also, they felt that there might be some problems applying it. Also, and they don't have enough information about contact lenses or medicated contact lenses. So that's clearly identified that it's quite important that we educate patients as well as healthcare professionals about these dosage forms if it came into the market in the coming few years. In terms of the views from the healthcare professionals, they felt that it's quite good to use eye drops, simply again because it's going to reduce the frequency and it increase the efficacy of using the contact lens, and as well as improving the adherence of the patient with using the eye or the ocular medication. However, they have some kind of reservations, and some of them said that the contact lenses will cause eye infection and eye inflammation. So this came up in 3/4 of the healthcare professionals that we surveyed. They said that they are worried because of the infection caused by the contact lenses. Also, it will be more expensive compared to the conventional dosage forms, and there will be some challenges counseling the patient on how to use them. And this was eye-opening for us. So when we realized that there might be infection issues, we decided to do another study and see if we can design a contact lens that reduce the bacterial adherence to their surface, and as a result of this, reduce the infection that is caused by contact lenses. One of the conditions that is quite common is called microbial keratitis, which is associated with the use of contact lenses, and this might be because of the bacteria that grow on the surface of these lenses, and as a result of this affect the patient eye. So in this study, what we did is we tried to design a lens that can resist the adherence of Pseudomonas aeruginosa, and this is one of the organisms that will cause microbial keratitis. So we designed our contact lenses, and what we did is we added 3 surfactants, so Bluxmar 188, Polysorbate, and then Tritionic 90R4. So we believe that these surfactants will prevent the adherence of bacteria because of their hydrophilic nature, or if you look at the structure of this surfactant, they will have hydrophilic moieties, and these moieties will prevent the adherence of bacteria or stop or reduce the adherence mechanism of this bacteria, and as a result of this, that will reduce the amount of bacteria that will grow in the surface of the contact lenses. So we based our studies on an assay, which is called bioluminescence assay. So basically, if you have bacteria, they have to produce some ATP to enable them to grow, and then the ATP in presence of luciferase will result in formation of AMP and then light. So this light, or the emitted light, will be measured at around 516 nanometer. And we found that the concentration of ATP, which reflected the number of bacteria that are growing in the surface of contact lenses, for example, will increase. If the ATP number increases, you can see that the light emission increase as well as per the concentration of ATP. So we designed our contact lenses, and we incorporated our surfactant, as I said. And then we started looking at the probabilities of these lenses. Majority of them had good light transmittance, apart from the Tritionic, which reduced the light transmittance to around 60 or 50%. So this means that we can't use them because they are very opaque, and then they will affect the vision of the patients. And then when we started looking at the amount of bacteria that grow or adhere to the surface of these design contact lenses, for example, when you use polysorbate at different concentrations, or using different kind of polymer for the manufacturing of the contact lenses, we found that the number of bacteria growing on this surface or adhering to the surface of the contact lens has dropped significantly when we added the non-ionic surfactant. Some of them managed to reduce the number for up to 6 hours, and then others managed to reduce the number of bacteria adhered on the surface up to 16 hours, as you can see in these cases. And again, because if you look at the chemical structure of these non-ionic surfactants, we have polyethylene oxide in the case of Bluxmar 188, and this hydrophilic, this chain, or this part of the chain which is hydrophilic, will inhibit the adherence of the bacteria, and as a result of this, you can design a contact lens which minimizes microbial keratitis in some cases. And then I'm going to end up with a bit of where we are in terms of contact lenses usage for ocular drug delivery. Over the last few months, I worked with an MPharm student, and we wanted to do a systematic review to see how contact lenses are used to deliver ocular drugs. So we looked at different databases, including PubMed, Science Direct, and Scopus, and Web of Science, and we looked over the last 10 years in terms of how these contact lenses were used to deliver ocular drug delivery system. And as you can see from this figure, the use of contact lenses to deliver drug has started some time ago, but it became very popular over the last 5 years. And then as you can see, these bars in dark blue, these are the studies that we included. So in total, we managed to include 124 studies that met the inclusion criteria. And looking at the therapeutic area where contact lenses were applied, we found that majority of the studies have looked at the antibacterial delivery using the contact lenses, and this was followed by beta blocker and then non-steroidal anti-inflammatory drugs, or NSAIDs. So these 3 areas was basically around 2/3 of all the published literature. Again, to reiterate, we said that the main advantage of using contact lenses is to sustain or control the drug release over a longer period of time. And this was reflected on the studies that we looked at. So a majority of these studies explored the controlled release of the drugs across the contact lenses matrix. Again, with contact lenses using to deliver the drug to the anterior part of the eye or the posterior part of the eye, almost 2/3 of the studies looked at interior part of the eye, and then 1/3 looked at the posterior segment of the eye. The kind of clinical studies that I looked at with the glaucoma, which is a majority of the studies, and then this was followed by the post-operative pain, and then conjunctivitis, which represented 18% of the included study. Okay, again, as part of where we are, so some of these medicated contact lenses, as we said, they offer good potential as a future drug delivery devices. So there are some clinical trials, and some of them has been successful through stage 1 or phase 1. So, for example, this was a study that has started in 2016 through the University of Florida, and they were looking at the effectiveness and safety of timolol and dorzolamide loaded contact lenses to be used for treatment of glaucoma. The study has not concluded yet, so I think they will finish recruiting and summarizing the data in November this year. And there was another study by a company called Mediprint. They designed contact lenses to deliver, again, drugs through the eye, and then they have been successful in phase 1 of their trials, and then they moving into phase 2 very soon. So in conclusion, so medicated contact lenses is a promising new dosage form that can be used for treatment of ocular conditions. Yet there are a number of challenges. It's quite important to investigate patient views about novel devices before large-scale manufacturing and marketing. Also, healthcare professionals can advise us about the usability of the design dosage form in clinical settings and flag up any possible challenges. And the reason why I'm saying these are important because maybe you have heard of the insulin inhalers that were developed back in 2007 by Pfizer, Zubair, for example. They didn't succeed, and then they have to withdraw from the market after 1 year, and simply because the patient decided not to use them because they were quite big in size, and then it wasn't user-friendly. Before I finish, I would like to acknowledge the contribution from my students in the labs, and then the collaboration that I did with Dr. Amr Elshaer from Moorfields Eye Hospital. And also thank you, and thanks Zubair and Richard for your kind invite for the meeting today. Thank you.
Thanks, Amr. That's a fantastic talk. Really exciting new developments, which certainly really encouraging. I'm sure that our patients will be really pleased to hear the potential for contact lens delivery over potentially eye drops. The first question from Zubair is the 8% reduction in light transmission after your NP treatment significant in terms of reducing quality of vision?
Not really. So I think anything below 80% will start affecting the vision of the patients. But ideally, we would like to have our contact lenses, including the drug, to be as clear as possible, and then has a high light transmittance. I think the challenge in the study that we did is we included nanoparticles, and then they can have an effect on the light transmittance. But if you have the drug suspended in the lens matrix, that wouldn't have that much effect in terms of the light transmittance. But if you would like to sustain the drug release longer, and as a result of this, you can include one of these approaches that we discussed, cyclodextrins or maybe a surfactant and so on, that will inevitably will have an effect on the amount of the light that coming through the lens.
Yeah, thanks. I'd say, just for context, Zubair, that the cornea scatters about 10% of incident light, so 8% is pretty good. And if you give somebody dry eye by giving them some eye drops, that's going to increase their light scattering. So next talk from Dan Nugent, wondering whether you can provide your thoughts on the change in the form of the drug from crystalline to amorphous after loading into the NP would have any impact on the activity or efficacy of the drug.
Yeah, thank you very much, Dan, for your question. So basically, the crystalline form of the drug doesn't have an effect on the drug in terms of its effectiveness or efficacy. But it will have an effect on the drug behavior. So, for example, we know that the crystalline form of the drug is more stable and it is less soluble. Okay, so when you put it in a solution, for example, it will take longer time to solubilize this crystalline form of the drug. So this means it will take longer time to have a therapeutic effect. And then that's why it's not so solubility of the solution will be the challenge in this case. But the amorphous form of the drug, because it doesn't have a well-defined crystalline shape, so they are much easier to solubilize, because when you put it in the solution, they are faster in terms of the solution, and then as a result of this, you will have a faster bioavailability compared to the crystalline form. Usually with the different techniques or different kind of pharmaceutical processing that we use in the lab. I think Raj in the last talk was talking about lyophilization and then the spray drying. So as part of this process, you have the crystalline form of the drug, and then you put it in a solution so that you lose the crystallinity of the drug, or the drug become amorphous. And then when you dry again, so it will remain in the amorphous form. Okay, so this is desirable when you would like to have a faster bioavailability. However, if you have a stability issue, it's less desirable, because this means that the drug will be less stable compared to its crystalline form, if this makes sense.
Okay, thank you. And there were some other parts of that question, but I think we're short of time, so apologies to Dan that we haven't got to all of those. Thank you very much, Amr. That was a fantastic talk. We'll now move on to our next speaker.